Hi Skylar, what's up?
Hi Ian, just got finished with my experiment on Drosophila melanogaster.
That's awesome. What was the experiment about?
Have a seat and I will explain. The purpose of this lab was to analyze the inheritance of specific genetic traits using D. melanogaster. Studying genetics is very important. The most important reason is to help understand human diseases. Understanding these diseases and diagnosing them early can allow people who suffer from the diseases to live a longer or better life. Another reason studying genetics is very important is it can help scientists figure out where humans came from. Studying genetics is very important in forensics, because DNA can be found at crime scenes and scientists can compare it to suspect DNA to see if they committed the crime.
So how do scientists study genetics?
Scientists study genetics using one of many model organisms. Model organisms are organisms that have a short life cycle, are inexpensive to care for and have similarities to humans. D. melanogaster are model organisms, because they have about a 10-day life cycle, they are inexpensive, they do not need a large area to be kept in, and they produce a large amount of offspring. D. melanogaster life cycle includes 4 stages: egg, larva, pupa and fly. D. melanogaster have been used for over 100 years, with the first documented experiment in 1901 by William Cassels' group. Thomas Hunt Morgan became the father of D. melanogaster research after he changed the theory of inheritance, saying what genes were and that they were found in chromosomes. One of the most recent experiments involving D. melanogaster was in 1995 when Christian Neuslein-Volhard, Eric Schaus, and Ed Lewis found that a lot of the genes that are used for fly development are the same genes that are needed for all animal development, even humans. In this study, B-type female flies with wings and white eyes were mated with wild-type male flies with wings and red eyes. This was done to see which traits were dominant and which were recessive, also to see if the traits were autosomal or sex-linked. Autosomal traits are passed down on any of the first 6 chromosomes for D. melanogaster that are called autosomes. Sex-linked traits are inherited on sex chromosomes.
What did you think was going to happen in this experiment?
I thought all of the progeny flies would have red eyes and wings, because I guess that red was dominant and that wings were dominant. I also thought that the traits would both be autosomal.
Very interesting. What materials and methods did you use in this experiment?
A microscope was used for this experiment to sex the flies. The flies were given fly nap, a small insect anesthetic, prior to sexing them. The flies were put onto a white note guard and put under the microscope to be sexed and separated by sex and eye color and then counted. A vial was prepared with 1 scoop of dry media, 1 scoop of water, then 5 grains of yeast. The fly-napped flies were put into the vial, then a sponge was put in the top of the vial and the vial was labeled. Each time the vial needed to be cleared, fly nap was inserted between the sponge and vial side until all of the flies stopped moving. Then the flies were removed, leaving the larva. It is important to note that the fly nap should not touch the sponge so the progeny are not killed.
So what were the results?
In this experiment, female B-types were crossed with male wild types. The progeny from these 2 parents were 30 out of 40 red eyes and 10 out of 40 white eyes. The 30 with red eyes were all female and the 10 with white eyes were all males. Then the 30 females with red eyes were crossed with the 10 males with white eyes to result in an F2 progeny with 26 out of 31 having red eyes and 15 out of 31 having white eyes. There were 18 females with red eyes and 8 males with red eyes. Of the 15 progeny with white eyes, there were 8 females and 7 males.
Fascinating. Do you know why these were the results?
The first progeny that had 30 out of 40 red eyes and 10 out of 40 white eyes, with all of the red eyes female and all of the white eyes male, showed that the eye allele was sex-linked and the red eye allele was dominant. This refuted half of our hypothesis that the trait was autosomal, but it supported our hypothesis that red eyes was the dominant allele. The male red-eyed wild type would have been dominant and the female white-eyed B-type homozygous recessive. The Punnett square for this would look like this. As seen in the Punnett square, the red eye allele had to be dominant, because the male red-eyed wild type only had a dominant allele to pass on to all the female progeny and only a Y to pass on to all of the male progeny, which resulted in all the female progeny being red-eyed. Since the female white-eyed B-type had only recessive alleles to pass on to all of the progeny. This also resulted in all of the males being white-eyed because they got the recessive allele from the mother and only Ys from the father. The I allele being sex-linked is also proven in the F2 progeny. The Punnett square for the cross of two F1 progeny would look like this. This also proves the I allele was sex-linked because, as seen in the Punnett square, the female progeny should have been half red-eyed and half white-eyed, and the male progeny should have also had half red-eyed and half white-eyed. The F2 male progeny were almost exactly half and half, since there were 8 males with red eyes and 7 males with red eyes. But the female F2 progeny had 18 red eyes and 8 white eyes, which is 70% red eyes and 30% white eyes. The reason this didn't show as exactly half and half was probably due to sample size. If the F2 progeny had a higher total, then it is likely that the males would even out to about 50% red-eyed and 50% white eyes. But then again, we were working with living organisms, so it is possible it would have never perfectly been half and half.
Wow, that's awesome.
Drosophila melanogaster were used in this experiment to analyze the inheritance of the I allele. This experiment showed that the I allele was sex-linked. Scientists everywhere used D. melanogaster because they are model organisms. Scientists have been using D. melanogaster for over 100 years, since they have such a short life cycle and are easy and inexpensive to care for. Scientists use D. melanogaster to study genetics, which is very important because it helps understand human diseases and how these diseases are passed on. It helps scientists figure out where humans came from, and it is very important in forensics.